Greetings, I am Mark Am, member of group number 6 of BIO149A31. Our group is tasked to explore and discuss recombinant DNA technologies. Recombinant DNA is an artificial form of DNA that cannot be found in natural organisms. It is made in the laboratory by joining together genes taken from different species. This is done by selecting and cutting out a gene at a specific point on a strand of a DNA using restriction enzymes which act as molecular scissors. The cornerstone of most molecular biology technologies is the gene. To facilitate the study of the genes, they can be isolated and amplified. One method of isolation and amplification of a gene is to clone the gene by inserting it into another DNA molecule that serves as a vehicle or vector that can be replicated. When these two DNAs of different origins are combined, the result is a recombinant DNA molecule. Although genetic processes such as crossing over technically produce recombinant DNA, the term is generally reserved for DNA molecules produced by joining segments derived from different biological sources. The recombinant DNA molecule is placed in a host cell. This host cell can be prokaryotic or eukaryotic. The host cell then replicates, producing a clone, and the vector with its foreign piece of DNA also replicates. The foreign DNA thus becomes amplified in number, and following its amplification can be purified for further analysis. This technology is important because it enables the creation of multiple copies of genes and the insertion of foreign genes into other organisms to give them new traits, such as antibiotic resistance or new color. One of the first ways in which the technology was deployed was to re-engineer microbial cells to produce foreign proteins. This facilitated the manufacture of human proteins on an unprecedented scale at minimum cost, thereby opening the way to study the function of proteins in greater detail and to their therapeutic use. By 2001, over 80 recombinant DNA-based products had been approved for treating diseases and for vaccination, and a further 350 recombinant DNA-based products were being tested for safety and efficacy. This technology is also an important tool in agriculture, being used to improve the plant's resistance to pests and to increase the crop's yield. In order for us to understand more about this recombinant DNA molecule, let us first explore its roots, where it began, and how it happened.
All organisms on Earth evolved from a common ancestor, so all organisms use DNA as their molecule of heredity. At the chemical level, DNA is the same whether it is taken from a microscopic bacterium or a blue whale. As a result, DNA from different organisms can be cut and pasted together, resulting in a recombinant DNA. The first recombinant DNA molecule was produced in 1972 by Stanford researcher Paul Berg. Berg joined together DNA fragments from two different viruses with the help of particular enzymes, restriction enzymes and ligase. Restriction enzymes are like molecular scissors that cut DNA at specific sequences. If the DNA from the different sources is cut within the same restriction enzyme, the cut ends can be joined together and then sealed into a continuous DNA strand by the enzyme ligase. In 1973, the first organism to cut recombinant DNA was engineered by Herb Boyle at UCSF and Stanley Cohen from Stanford University. Together, they introduced an antibiotic resistance gene into E. coli bacteria. Notably, they also produced bacteria that contain genes from the toad Xenophus lapis, which showed DNA from different species to be spliced together. Paul Berg was awarded the 1980 Nobel Prize in Chemistry for his fundamental studies of biochemistry of nucleic acid with particular regard to recombinant DNA. The ability to cut, paste, and copy molecules of DNA was not only a watershed moment for scientific research, but spawned an entire industry built on genetic engineering. Genentech, the first biotechnology company, was founded by Herb Boyer in 1976. By 1982, the FDA approved Genentech's first successful product, a synthetic form of human insulin produced by bacteria that were engineered to contain the insulin gene. Today, recombinant DNA technology is used extensively in research laboratories worldwide to explore myriad questions about gene structure, function, expression pattern, regulation, and much more. One widely used application involves genetically engineering knockout animals, typically mice, to contain a non-functional form of particular gene of interest. The goal of such experiments is to determine gene function by analyzing the consequences of missing gene. While knockout mice are generated to answer questions in many different fields, they are particularly used in development biology and have led to an understanding of some of the essential genes involved in the development of an organism from a single fertilized egg. Recombinant DNA techniques are also a cornerstone of the biotechnology industry. One example of the generation of genetically engineered plants to produce an insect toxin called Bt toxin. The Bt gene is derived from a bacterium called Bacillus thuringiensis and produces a toxin that disrupts gut function in the larvae or caterpillars of certain insects that are crop pests. The gene that produces Bt toxin is introduced into such plants by recombinant DNA technology and results in the selective killing of crop-feeding insects. This development has had a major economic impact and reduced the expenses of pesticides used per year and has increased the life and success of several crops. How does recombinant DNA technology work? The organism under study, which will be used to donate DNA for the analysis, is called the donor organism. The basic procedure is to extract and cut up DNA from a donor genome into fragments containing from one to several genes and allow these fragments to insert themselves individually into opened-up small, autonomously replicating DNA molecules such as bacterial plasmids. These small circular molecules act as carriers or vectors for the DNA fragments. The vector molecules with their inserts are called recombinant DNA because they consist of noble combinations of DNA from the donor genome, which can be from any organism, with vector DNA from a completely different source, generally a bacteria, plasmid, or a virus. The recombinant DNA mixture is then used to transform bacterial cells, and it is common for a single recombinant vector molecule to find their way into a different individual bacterial cell. Bacterial cells are plated and allowed to grow into colonies. An individual transformed cell with a single recombinant vector will divide into a colony with millions of cells, all carrying the same recombinant vector. Therefore, an individual colony contains a very large population of identical DNA inserts, and this population is called DNA cloning. A great deal of analysis of the cloned DNA fragment can be performed at the stage when it is in the bacterial host. Later, however, it is often desirable to reintroduce the cloned DNA back into the cells of the original donor organism to carry out specific manipulations of genome structure and function. Hence, the protocol is often as follows. First, identify donor gene of interest by using process. Then, clone donor gene of interest in the bacterium. Then, characterize donor gene in bacterial system. Modify donor gene, and lastly, reintroduce donor gene into donor cells.
These are the 4 basic tools of the recombinant DNA transfer process. Number 1 are enzymes. Enzymes are biological macromolecules that act as specific catalysts during biological processes. They are very sensitive to changes in the chemical and physical environment. That is to say, each enzyme accelerates one or more specific chemical reactions without affecting the final equilibrium concentration of reactants and products. In addition, the enzyme is not broken down during the reaction it catalyzes. In the thermodynamic language, enzymes reduce the activation energy of a reaction but do not affect the free energy change of the overall reaction. Many enzymes are so effective that they will catalyze intracellular reactions which are too slow to be observed at all under comparable conditions in the absence of enzyme catalysis. Enzymes make and break covalent bond in a cell. In general, an enzyme regulates the biochemical reaction pathway. The next type of tool are vectors. A vector acts as a template for the cloning of a specific gene. This gene needs to be inserted into the vector by a recombinant DNA. before the vector can be inserted into a target cell host. These are essential components for recombinant DNA technology. They act as the vehicle to carry recombinant DNA into the target host cell. Vectors have special restriction sites onto which the recombinant DNA can insert itself. They are relatively easy to manipulate and introduce into the target host cell. Another important property of vectors is that they can be efficient origin of replication. The most common types of vectors are plasmid, viruses, or cosmids, and these can be linear or circular and is always double-stranded. There are 4 types of enzymes. The first type is the restriction endonuclease. It acts as a molecular scissor to cleave the DNA at a specific sequence. The most common type is the endonuclease type 2. It recognizes 4 to 8 palindromic sequences. Different endonucleases will have a different way of cleaving the DNA. There are 2 types: the asymmetrical cleavage, which leaves either 5 or 3 sticky ends. The other type is the symmetrical cleavage. It leaves a blunt end. The second is the DNA ligase. This enzyme is responsible for joining fragments of DNA together by reforming sugar phosphate backbone. The third is the Taq polymerase. It is an enzyme that is used during PCR to amplify copies of gene. It is stable at high temperatures required for PCR to take place. And the last is reverse transcriptase. It is an enzyme that is used to convert mRNA back to cDNA. The general requirements for vectors. They should contain unique restriction sites that act as an attachment site for the new DNA. It must contain efficient origin of replication. It can be introduced easily to the host cells. It must contain genes that allow for selections, such as antibiotic resistance, and it must contain expression factors. The most commonly used vectors are plasmids, bacteriophages, and plasmids cosmids. These are the hybrid of plasmids and bacteriophage. The third tool are the DNA and the mRNA. We can use either of the molecules as source for the gene of interest. DNA as a source is isolated from lysed cells. It is then separated and partially cleaved. mRNA as a source is transcribed back to the DNA using reverse transcriptase. The cDNA is then being referred to as the cDNA library. The advantages of using cDNA is that there is no longer any intron in the DNA, so we won't introduce truncated proteins. The last tool are cells. Cells are a fundamental component of all biological existence, often referred to as the building blocks of life. Some organisms, such as bacteria, are unicellular. Cells exist as independent units of life. Other higher organisms are multicellular, comprised of many cells. Within these multicellular organisms, groups of specialized cells collaborate to perform specific functions. All cells adhere to specific fundamental principles, irrespective of their eukaryotic or prokaryotic classification. All cells must have a plasma membrane to separate the intracellular space from the extracellular space, contain heritable genetic material to ensure the propagation of the species, contain intracellular compartments to regionalize specific functions, regulate their osmotic gradient via the transformation of chemical species across their membrane.
Principles of recombinant DNA technology, 4 steps. The first step is gene cloning and development of recombinant DNA. Any gene to be cloned must be inserted in a cloning vector. A foreign gene introduced into a bacterium cell will not be replicated in bacteria. The reason for this is that the enzyme DNA polymerase is responsible for copying DNA. It does not initiate the process at random. It is initiated at selected sites known as origin of replication. Generally, small fragments of DNA do not possess an origin of replication. Using our DNA technology, it is possible to insert the gene into a cloning vector, which in turn will make copies of fragments. A cloning vector is simply a DNA molecule possessing an origin of replication, and which can replicate in the host cell of choice. Most commonly, plasmids, extrachromosomes, anonymously replicating circular DNA molecules are used as vectors. Sometimes, viruses are used as vectors for gene insertion into microorganisms, but they are better vectors for animal cells. Cutting and insertion of desired foreign gene into the plasmid requires special enzymes known as restriction endonuclease or restriction enzyme. These enzymes cut large DNA molecules into shorter fragments by cleavage of the specific nucleotide sequence. called recognition sites. Therefore, restriction endonucleases are highly specific deoxyribonucleases, or DNAs. Both vector DNA and foreign DNA to be inserted in cut by the same restriction enzyme, generating complementary ends. Thus, ends of foreign DNA make perfect match with cut ends of vector, and to join make again a circular molecule. This process can be understood in detail by making an example of PBr322 plasmid. It is commonly used cloning vector. Firstly, purified, closed, circular PBr22 molecules are cut with a restriction enzyme that lies within either of the antibiotic-resistant genes and cleavage the plasmid DNA only once to create a single, linear stick-ended DNA molecules. These linear molecules are combined with prepared target DNA from the source organism. This DNA is cut with the same restriction enzyme which generates the same sticky ends as those in plasmid DNA. The DNA mixture is treated with T4 ligase in the presence of ATR under these conditions. A number of different ligase combinations are produced, including the original closed circular plasmid DNA. To reduce the amounts of these particular unwanted ligation products, the cleavage in plasmid DNA preparation is treated with the enzyme alkaline phosphatase, trimming the 5 phosphate groups from the linearized plasmid DNA. Due to this T4 DNA ligase cannot join the ends of the phosphorylated linear plasmid DNAs. However, the two phosphodiester bonds that are formed by T4 DNA ligase after the ligation and circulation of alkaline phosphate-treated plasmid DNA with restriction to endonuclease digested source DNA are sufficient to hold both molecules together. Despite the presence of two nicks after the transformation, these nicks are sealed by the host cell DNA ligase. In addition, fragments from the source DNA are also joined to each other by T4 DNA ligase. The second step of the principle is transfer of vector into the host. The next step is a recombinant DNA experiment requires the uptake by E. coli of the RNA. The process of introducing purified DNA into a bacterial cell is called transformation. This is carried out by treating cells with calcium chloride and high temperature. A few transformed cells are obtained by this method. Extrachromosal DNA that lacks an origin of replication cannot be maintained within a bacterial cell. Thus, uptake of non-plasmid DNA is of no significance in the recombinant DNA experiments. Suitable strain of E. coli is used which lack capabilities to destroy plasmid DNAs, for carrying out exchanges between DNA molecules.
For the third process, selection of transformed cells. After transformation, it is necessary to identify the cells that contain plasmid colon DNA constructs. In PBr322, in which target DNA was inserted onto the BAM-hide site, recombinant bacteria are selected. All cells are grown successively on media containing antibiotic, ampicillin, or tetracycline, and cells showing the recombinant DNA, depending upon the restriction and enzyme site and loss of particular antibiotic resistance due to disruption of the gene. Selected recombinant bacteria are grown in a bioreactor to obtain the gene product. Other methods for selection of recombinant bacteria are, if synthesized gene is used, selection is easy as compared to DNA fragments used from genomic library of an organism, which requires selection for a suitable character. Second, by nucleic acid hybridization technique. All the steps are required to be considered carefully in making a recombinant bacterium. Prokaryotic organism E. coli is the most frequently used bacterium for recombinant technology. For the fourth step, transcription and translation of inserted gene. Transcription of DNA into mRNA is mediated through the enzyme RNA polymerase, which recognizes the binding site of DNA called promoter. The process of mRNA synthesis is terminated by a termination signal. This means only gene lying between promoter and terminator will be transcribed. Gene isolated in certain ways such as cDNA cloning or artificial synthesis do not have their own promoter. Therefore, they may be inserted into a vector close to promoter site. Even if a cloned gene carries its own promoter, this promoter may not function in the new host cell. In such circumstances, the original promoter has to be replaced. The position of cloned insulin gene with lac promoter in the vector is given in the figure 14.11. This gene is transcribed in the presence of lactose because lac operon functions in the presence of lactose and transcribe the gene attached downstream to attach it. Similarly, positions of ribosome binding site and termination codon are shown in the figure in respect to the insulin gene in the vector, making recombinant DNA. For an overview, first, isolate DNA. Second, cut with restriction enzymes. Third, ligate into cloning vector. Fourth, transform recombinant DNA molecule into the host cell. And fifth, transform cells will divide. Isolating DNA. First, crude isolation of donor DNA is accomplished by isolating cells, followed by disrupting lipid membranes with detergents, destroying proteins with phenol, degrading RNAs with RNase, and leaving DNA at the end. Crude isolation of plasmid vector DNA is accomplished by an alkaline lysis procedure or by boiling cells, which removes bacterial chromosomal DNA from plasmid DNA. To get pure DNA from either one or two, crude DNA is fractioned CSCl2 precipitated with ethanol, poured over a resin column that specifically binds DNA. Cutting DNA. DNA can be cut into large fragments by mechanical shearing. Restriction enzymes are the scissors of molecular genetics. Restriction enzymes are endonucleases that will recognize specific nucleotide sequences in the DNA and break the DNA change at those points. A variety of RE have been isolated and are commercially available. Most cut at specific palindromic sites in the DNA. These cuts can be staggered with generic stick or overhanging ends, or a blunt which generate flash ends. Joining DNA. Once you have isolated and cut the donor and vector DNAs, they must be joined together. The DNAs are mixed together in a tube. If both have been cut with the same RE, the ends will match up because they are sticky. DNA ligase is the glue of molecular genetics that holds the ends of the DNAs together. DNA ligase creates a phosphodiester bond between two DNA ends.
Amplifying recombinant DNA. To recover the large amount of recombinant DNA molecule must be amplified. This is accomplished by transforming the recombinant DNA into a bacterial host. Once in a cell, recombinant DNA will be replicated. When the cell divides, the replicated recombinant molecules go to multiple other cells, which themselves will divide later. Thus, DNA is amplified. Vectors. The requirement for cloning a vector includes the following: should be capable of replicating in host cell, should have convenient RE sites for inserting DNA of interest, should have a selectable marker to indicate which host cell received recombinant DNA molecule, and should be small and easy to isolate. Bacterial plasmids are small circular DNA molecules that are separate from the rest of the chromosome. They replicate independently of the bacterial chromosome. Useful for cloning DNA inserts less than 20 kilobase pairs. Inserts larger than 20 kilobase pairs are lost easily in the bacterial cell. Bacteriophage lambda contains several regions of 15 kilobase pairs that is not required for replication or formation of Hodgkin's phage in E. coli. Thus, lambda can be used as cloning vector by replacing the central 15 kilobase pair with 10 to 15 kilobase pair of foreign DNA. Cosmids are hybrids of phages and plasmids that carry DNA fragments up to 45 kilobase. They can replicate like plasmids but can be packaged like phage lambda. Expression vectors are vectors that can carry host signals to facilitate the transcription and translation of an inserted gene. They are very useful for expressing eukaryotic gene in bacteria. Yeast artificially chromosomes, or YACS, are yeast vectors that have been engineered to contain centromere, telomere, or origin of replication, and a selectable marker. They can carry up to 1,000 kilobase of DNA. Since they are maintained in yeast or eukaryote, they are useful for cloning eukaryotic genes that contains introns. Also, eukaryotic genes are more easily expressed in a eukaryotic host, such as yeast. Bacterial artificial chromosomes, or BACS, are bacterial plasmids derived from ectoplasmids. They are capable of carrying up to 300 kilobase of DNA.
Applications of recombinant DNA technology. Its first application is in crop improvement. Genetic engineering has several potential applications in crop improvement, such as number 1, distant hybridization. With the advancement of genetic engineering, it is now possible to transfer genes between distantly related species. The barriers of gene transfer between species or even genera have been overcome. The desirable genes can now be transferred even from lower organisms to higher organisms through recombinant DNA technology. Second is the development of transgenic plants. Genetically transformed plants which contain foreign genes are called transgenic plants. Resistance to diseases, insects and pests, herbicides, drought, metal toxicity tolerance, induction of male sterility for plant breeding purpose, and improvement of quality can be achieved through this recombinant DNA technology. PT cotton resistant to bollworms is a glaring example. The third is development of root nodules in cereal crops. Leguminous plants have root nodules which contain nitrogen-fixing bacteria, the rhizopule. This bacteria converts the free atmospheric nitrogen into nitrates in the root nodules. The bacterial genes responsible for this nitrogen fixation can be transferred now to cereal crops like wheat, rice, maize, barley, etc. through the techniques of genetic engineering, thus making these crops too capable of fixing atmospheric nitrogen. The second application is in medicines. Biotechnology, especially genetic engineering, plays an important role in the production of antibiotics, hormone insulin, vaccines, interferon, enzymes, gene therapy, solution of disputed parentage, diagnosis of diseases, and the production of transgenic animals. And the third one is industrial applications. In industries, recombinant DNA will help in the production of chemical compounds of commercial importance, improvement of existing fermentation processes, and the production of proteins from waste. This can be achieved by developing more efficient strains of microorganisms. Specially developed microorganisms may be used even to clean up the pollutants. Thus, biotechnology, especially the recombinant DNA technology, has many useful applications in crop improvement, medicines, and industry.